The last type of membrane transport is active transport. And active transport is very different from passive transport in that, first, it does not rely on the process of diffusion. As a matter of fact, it moves things in the opposite direction, so against the concentration gradient. And in order to do that, active transport requires ATP. ATP is the energy currency of the cell. We take in all kinds of energy-rich molecules like carbohydrates and fats. We don't generate a lot of energy from proteins, but we certainly take in enough carbohydrates and fats to fuel our cells and keep ourselves alive. We cannot directly apply the energy found in the carbon-hydrogen bonds of carbohydrates to any kind of chemical reaction or to any energy-requiring process. Instead, we have to take all these different forms of energy and convert them into one common form, which is ATP. It's like if you had 3 different jobs and they all paid you in dollars. You're getting all this payment in one currency, even though it's coming from multiple sources. Then you have all these multiple things you have to pay, different people and businesses you have to pay, but they all accept a common form of money. That's the case with energy requiring processes in the cell. All those processes take ATP as the form of energy. ATP is actually a nucleotide. And you might remember a nucleotide has a sugar, phosphate, and base. ATP is an adenine-based base, and this is an RNA base. But nucleotides aren't just used for building nucleic acids. This is a nucleotide with 3 phosphate groups. 3 negatively charged functional groups are held together by covalent bonds. And it's sort of like holding together 3 magnets, or 2 magnets, I should say, and trying to push them together, even though the 2 negative ends are being pushed together. They don't want to hang out together. Only things of opposite charge want to hang out together. So in the covalent bonds that are holding these phosphate groups together, there's lots and lots of energy. These phosphate groups are just dying to break off. What we do in our cells is we take an energy-rich molecule like glucose, we go through cellular respiration and extract energy from that molecule and use it to drive the bonding between phosphate groups on ATP. So we basically convert glucose energy to ATP energy. When ATP is used to do something, it involves releasing one of those phosphate groups, and that's considered a release of energy. Actually, the release of energy is a transfer of a phosphate group to another molecule. So, we're going to look at a specific type of transport protein that moves solute from an area where it's in low concentration to where it's in an area where it's in greater concentration. ATP is going to be used to force that to happen. ATP will donate a phosphate group to this molecule, and when the phosphate group attaches, it actually changes the protein's shape. Because remember, the protein is all held together by covalent, ionic, and hydrogen bonds between the R groups of amino acids. Once you stick a big old negatively charged phosphate group on that protein, that screws up all the interactions that have held together the tertiary structure, and the shape changes. When the shape changes, that forces these substances that have bound into the protein to the opposite side of the membrane. That shape change kicks off the phosphate so that the protein gets back to its original shape. Let me show you an example. The sodium-potassium pump. This pump, which sounds like it requires energy, and it does require ATP, this pump moves ions, potassium ions and sodium ions, against the concentration gradient. The pump is a protein whose sodium binding sites are facing the inside of the cell. And there are lots of cells that are constantly trying to create a high concentration of sodium inside of the cell, or outside of the cell, and potassium inside of the cell. If the concentration is high out here of sodium, then it's going to be hard to get sodium to move continue to move outside of the cell, so this pump is used. The binding sites for sodium are facing inside the cell, and sodium can freely bind, no matter what the concentration is. Once it binds, it opens up a site for ATP. And ATP binds to the protein. A phosphate group is released and binds to the protein, which screws up the tertiary structure of that protein. The sodiums are actually kicked out, but the only place for them to go is out of the cell. Sodium is released outside of the cell, and now we've moved sodium up the concentration gradient. When this binding site is, or when this release site is open, that reveals binding sites for potassium, and potassium binds. Potassium binding causes the release of the phosphate group. We get another shape change, and potassium is released inside of the cell. And now we've moved potassium against the concentration gradient. There are cellular processes that require a high concentration of different ions on either side of the membrane. And this is just a really classic example of active transport. There are other types of active transport, other things that require energy. One I want to talk about is bringing extracellular material inside of the cell. The first type of endocytosis, which is what we're talking about, is called phagocytosis. And this is when cells make their pseudopods to wrap around some kind of food particle, or I think in one lecture I described white blood cells as wrapping themselves around bacterial cells. And the phagocytosis results in the creation of something like a transport vesicle. Here it's called a food vacuole. But it's a vesicle, membrane-bound vesicle, that's going to fuse with the lysosome so that this food particle can be further digested. Another type is penocytosis, which is described as cellular drinking. All cells occasionally pinch off extracellular environment to potentially get nutrients and precursor molecules for biological reactions. Again, a vesicle is produced which fuses with the lysosome. Last is receptor-mediated endocytosis. On the outside of cells, there are receptors that are there because this particular cell has to bind some molecule that may be in the extracellular fluid. Once the molecule binds, then a pit is produced. This is called a coated pit because there's a coat protein. And the coat protein makes this receptor specific for the material that binds the receptor. Once it binds and the pit is formed, it continues to pinch off until you get a vesicle that is coated and is targeted to the lysosome for joining with the lysosome and then release of the material. An example of this is how we bring cholesterol into our cells. We bring cholesterol in by receptor-mediated endocytosis. Like I've told you before, our cells need cholesterol. And in the bloodstream, cholesterol is carried by low-density lipoprotein, or LDL complexes. And LDL is just cholesterol and proteins, and it's just the way that this can be carried through our blood. We have receptors on the outside of our cells called the LDL receptors, and LDL binds to the receptor. Once that happens, then the LDL with its cholesterol is brought into the cell and fuses with the lysosome. Within the lysosome, the LDL is released, and the protein and cholesterol separated from each other. Ultimately, the cell will use these amino acids from the proteins and the cholesterol that's been carried, and the receptor is actually going to be recycled back up to the cell surface. One of the causes of high cholesterol, especially that really early onset high cholesterol, or familial, runs in the family, hypercholesterolemia, is when people lack the receptors on cells, or they have a low number of receptors on cells for LDL. LDL then is not picked up by cells, which actually isn't such a big deal because cells can make their own cholesterol, But what is a big deal is that LDL remains in the blood, and it collects and causes plaques, causes blockage in veins and arteries. So there's an example of real live cell use of receptor-mediated endocytosis. So overall, I want to summarize the difference between passive transport and active transport. Passive transport requires no energy, and active transport requires energy. This is a movement against the concentration gradient, and passive transport is a movement down the concentration gradient. Now within passive transport, remember there are two types. There's diffusion and there's facilitated diffusion, but both are diffusion. Osmosis is really a special form of diffusion, not facilitated, but it's a movement of water down the concentration gradient, and a movement of water from hypotonic solution to hypertonic solution. All of these are examples of passive transport. It's just that sometimes proteins are required for help, but for other substances, proteins are not required. I want you guys to take a few minutes, push pause on your slideshow or on your video here, and fill in the squares regarding transport across the membrane. Molecules cross cell membranes by either passive transport or active transport. Active transport requires ATP and uses membrane proteins. In active transport, we're moving substances against the concentration gradient, while in passive transport, we're moving substances down the concentration gradient. Passive transport can either be regular simple diffusion or facilitated diffusion, and if it's facilitated, it also uses membrane proteins. Simple diffusion is the process by which water, gases, and nonpolar substances cross the membrane. Facilitated diffusion is what's required for the movement of polar molecules and ions, anything that is basically large and hydrophilic.